Hello, I'm Bretton, one of our MCAT tutors here at Inspire Advantage, where we help students get into medical schools and other professional programs. And welcome back to another episode of MCAT Bites. Today we'll be discussing oogenesis, or oogenesis, the process of egg cell development in the ovaries. Understanding oogenesis is crucial for the MCAT, as it's a fundamental concept in reproductive biology. You can expect to see at least a few questions on the Biochem section about oogenesis. So this is definitely something you'll want to add to your Anki cards. First, let's start off by defining oogenesis. Oogenesis is the process by which a diploid germ cell undergoes meiosis to produce haploid egg cells, also known as ova. This process begins at birth and continues throughout a woman's reproductive years. Let's break down some of these big words real quick. A germ cell just means it's a sex cell. It's either a sperm or an egg. And diploid just means it's 2N, meaning it has two sets of chromosomes. And eventually that's going to divide into just a single N. So let's start at the beginning here for oogenesis. We begin with a diploid germ cell called the oogonium. That's this guy. And we know it's diploid because it's got 2N, two sets of chromosomes. During fetal development, oogonia undergo mitosis to first increase the numbers. So that's all that's going there, is we're just going to make a bunch of these. Around 5 months of fetal development, the oogonia enter meiosis I and become primary oocytes. However, these primary oocytes pause in prophase until puberty. And this is something the MCAT loves testing. You need to remember that the primary oocyte is paused in meiosis I in prophase. You'll hear them call it pause or arrested. So once a woman hits puberty, a group of primary oocytes will resume meiosis I each menstrual cycle. The primary oocyte, this guy here, is going to divide unequally into a primary polar body and a secondary oocyte, and a small polar body. The secondary oocyte will then begin meiosis II, but pauses in metaphase II until fertilization will occur, if it occurs. And what we can see is that by the end of all the divisions, we're going to have 3 polar bodies, and these are going to do basically absolutely nothing. It's only the ovum that could actually become fertilized to make a zygote. The resumption of meiosis I in the primary oocyte is triggered by a surge in something called luteinizing hormone, shown here as LH, during the menstrual cycle. The LH surge occurs around day 14 of a typical 28-day menstrual cycle, coinciding with ovulation. Follicle-stimulating hormone, FSH, also plays a role in stimulating growth and maturation of follicles containing the oocytes. The interplay between these hormones and the developing follicles is essential for proper progression of oogenesis and the menstrual cycle. If fertilization occurs, the secondary oocyte completes meiosis II, producing a mature ovum and another polar body. The polar bodies eventually will degenerate, while the mature ovum can be fertilized by a sperm cell to form a zygote. And if you've taken a plant biology course, this might be kind of similar to what you've learned about how plants divide. So I've just shown that here up top. So this isn't going on in humans, this is going on in plants. But just like us, they're kind of making these 3 little polar bodies that in plants are just used for nutrients. Humans, a little bit of nutrients, but mostly waste, we think. Now let's finish off with just a quick little practice problem to make sure that you're retaining important MCAT info. If a primary oocyte has 46 chromosomes, how many chromosomes will the mature ovum have after completing meiosis II? Take a few moments to think about this. To solve this problem, we need to remember that meiosis consists of 2 divisions, and each division halves the number of chromosomes. So if we start with a primary oocyte with 46 chromosomes, after meiosis I, we'll have 23 chromosomes. After meiosis II, the mature ovum will also have 23 chromosomes. Therefore, our answer here will be 23 chromosomes. Oogenesis is the process of egg cell development, beginning with diploid oogonia and ending with haploid mature ova. The process involves meiosis with a pause/arrest in prophase I and results in the production of polar bodies. The resumption of meiosis I in primary oocytes is triggered by the LH surge during the menstrual cycle, with FSH also playing a role in follicle development. We'll explore this in a lot more detail in a future video. Understanding the key stages, chromosome number, and hormonal regulation of oogenesis is absolutely essential for success on the MCAT. Watch our next video on hormonal regulation, but this one I want you to really take away the role of LH, FSH, and arresting that happens in prophase I. That's all for today's MCAT Bite on oogenesis. Thank you so much for watching, and I'll see you next time.